Okay, well, thanks for people joining for another week. I wanted to first bring up this meeting next Friday, Thursday and Friday is happening. I mentioned it last week. Just to give you a little background for people who don't know, I had approached Dr. Kashyap Patel, who's CEO, president of some of these nonprofits. One nonprofit he runs is NOLA. No one, didn't exactly stand for. I forgot. Yeah, there's no one left behind or something like that. Anyway, so his group is dealing with cancer. No one left alone. Sorry, that's what it is. No one left alone. But his focus. He's an oncologist, and he's focusing on cancer and the impact as nonprofits or big patient advocacy groups, and he's doing a lot of good work at promoting research and getting money to do nice work with research and cancer research in the Carolina areas and other places. But he had released he had done an interview in a little article in one of these journals that do these kind of articles on his observation, and was concerned. He started noticing in the past year or 2 years a spike in different types of cancers, not just in the U.S. but all around the world, that shouldn't be happening pre-COVID. So he was really concerned, or he is really concerned, the fact that he thinks there's a link between SARS-CoV-2 maybe potentially perpetuating rapid increase of cancers that are occurring that shouldn't be. But he had at that point he's just looking at observational thing, and he wants data to actually show if it's true one way or another. So I had approached them because we had maybe potential observations or data in the acute phase to show that it could potentially happen, with cancer link, but we don't have any validation of this. It's just basically looking at the data saying potentially it could be a link, or maybe these factors that come up early on in COVID go away and they don't cause cancer. So when I approached him, he got excited when he saw that, because he has the endpoint data, and he said, let's do this nice meeting here next week. He really quickly, I invited some people from this group to here, and then he invited a bunch of folks from different places that you see here. These are some of the people who are coming from different areas, biotech, universities, and some government officials showing up. And the whole goal is to bring awareness to it, and potentially maybe some of these folks might want to provide funds for us to actually show one way or the other if SARS-CoV-2 does actually cause cancer. And if it does, then we should be ahead of it before it becomes a huge problem. Or if it is going to become a huge problem, how do we tackle that problem before it's too late? So that's the whole goal of this. There will be online attendance if you want, so if you can attend in person, contact. I'll send these slides for folks too, but you could always email me. Contact Winnie Miente, and she'll give you a Zoom link that people can attend online. But I think this would be a nice meeting to have discussion about this, and potentially maybe funds to be generated to research this one way or the other. But yeah, so people want to attend. It's next Friday's the meeting for the online part.
Okay.
Well, I guess this leads to planning in-person meeting, but maybe that's for future. But for papers, and there's been, I don't think, any new papers coming out yet. I know there's a couple of papers under review. Our mitochondrial paper hopefully will be accepted soon. But for updates, yeah, it's basically the same updates as last week. I know Al's been working on the long COVID paper with some people in this group. It's a case report, a couple of case reports that are interesting. So there's a lot of data there the patients had consented and agreed for us to work on. So if people want to help, the more the merrier. Reach out to me or Al, and we could get you on board. Right now, we're at the point of we have to pile through the data that on this one or a couple of the patients there to actually see if we could notice any trends, or work on this paper to bring in insight on these interesting case reports. Okay, so the COVID news. So there's I haven't seen too many papers pop up this week on COVID, long COVID. There's actually some interesting papers I'll get into, but one thing people weren't aware of, there's actually if you have this type of antigen home tests from Roche or SD Biosensor, you might want to go to this site here in the FDA, check the lot number. If you have that lot number, you should probably throw it out. But bring some awareness, because they think there could be some bacterial contamination in some of these tests. So if you have that, I would advise or share it with your friends and family to go to this FDA site, look over the lot numbers you might have, and if you have one of those, throw them out. Just bring awareness there if you haven't seen that. This article came out this morning. I guess WHO has declared now end for COVID global health emergencies based on the trends. I'm not sure if we should end it, but that could be a debate we could have outside of this. But I guess they did that. So one of the things we've been seeing over the past couple of months is a decline in the accuracy of the case reporting because a lot of the trackers are being shut down. There's no more funds to like push them forward, unfortunately. So that's a little disappointing to see, but there's still data luckily being generated that we could play with. This interesting article in Nature editorial came out a few days ago, and it seems like there's some background noise. If you could mute your microphone, that'd be good. But what's interesting here is that is what we're saying. Like before, we see these periodic spikes happening, and new variants come, there'll be a new spike and a new one. Now, what they discussed here, talking with several scientists in the field, interviewing them, they're saying it's probably going to be mini waves from now on, rather than seasonal surges that what people are predicting. And we're seeing that now with like the XBB.1.16. It really hasn't spiked. We'll go over that in a second, as you might expect, but it's more like little pockets of it's going up and then it comes back down, since it's the mutations that have occurred in XBB.1.16 and XBB.1.91 are not too far apart for those. And also, it's not the symptoms and the mutations that occurred don't seem to be causing too many other symptoms, that our XBB.1.6 that is taking over everywhere has been symptom-wise, there hasn't been anything too different other than what we talked about last week. That there seems to be a higher case of conjunctivitis or pink eye caused by this, but that was the only really new symptom that's seen. Mortalities or hospitalizations haven't gone up based on these new variants, which is a good sign. But it's only time can tell what happens if another Omicron type variant pops up soon or not. This is probably, I just thought I'd put this up here since I know a lot of folks in the field use Gessaid. You probably might have heard about this. Some controversy came up over the questionable behavior of Gessaid and some of the data being used and compliance issues. So I won't go too much into the detail here, but people interested, this is kind of goes over, it's the summary of what the issues are. A little background on Gessaid if you don't know Gessaid, but I'm sure everyone here is very familiar with it now. And also what occurs. I put some points here that discuss that the scientific community has been now uncovering and complaining about, saying, for example, Gessaid lacks mechanisms to move data from data to open access, which has been one of the major flaws that they never fixed. They control the data and sometimes arbitrarily give access or restrict access to folks, and then some of the compliance issues are of concern. So,
did they actually give examples? Because that sort of seems a very sort of vague comment. The way Gessaid controls access for different users seems to be arbitrary. I mean, it really needs a little bit more information there to help people understand what the real concern is.
Yeah, they taught the details in this article. I didn't put it here, but like for example, one of the details they gave, Gessaid although Gessaid made a comment saying they don't do this, but there was a science article that came out, I think, a couple of weeks ago in this, or last week, that actually said, for example, when a scientist was critical about Gessaid, they restricted their access, and they gave a couple examples of that, which is a retaliatory kind of thing that people are saying this shouldn't happen, of course, and then Gessaid, of course, refused that. So that's one example that was causing issue, and in that, I guess, original science, was it a science article that was, I think, they gave some pretty good examples of that. And then applying like sanctions to users. I mean, one of the main complaints is that the person who's running Gessaid is a finance guy and seems to have full control over it. And one of the issues is this open access thing. Like the data comes in there, but then it never gets released to open access databases. And then if you have full freedom to go to another database and send your data and submit your data there. Most people don't have time to do multiple submissions to going through all the processes. So that's one of the other main criticisms that I guess I've read.
But I think USAID has proven itself to be critical because it was able to move quicker to enable scientists to have a repository for findings for COVID that moved in a far quicker time frame than throwing it to NCBI.
Right.
The reason for this is that at the beginning, this is where people were able to quickly share data. It may have grown too big from what it originally was, but originally this was where you'd go for the latest COVID sequence data versus going to NCBI or any of the other places. Because NCBI had some pain in the arse routines that you had to go through as well.
Yeah, originally this was set up for influenza too, so that I guess one thing they discussed exactly what you said, Rob, where maybe they weren't prepared for the amount of data they got, and how to handle that, and they didn't change any of their policies along the way.
No, but it was easier to throw data to GISA than it was to NIH and to dbGaP, or was it the SRA data sets, because they had a whole load of requirements and stringencies to go through, whereas this enabled people to get the information out quick.
Right, exactly.
I'm not condoning if they're blocking people who criticize its stuff, but with regard to moving the data to open access, I think most people threw here as a stopgap to them submitting their full sequence information to NIH or other international open source databases.
I hear you. I agree. But I guess that's just the controversy around it now, and I guess it's enough. There's a lot more detail here that I'm not touching on. I have to go back and remind myself of. But I guess this is rubbing a lot of scientists the wrong way. That's the and then whether I guess GISA made a comment here in the Type article that they're looking into these complaints and they're going to try to address them. So I guess we'll see what happens in a month if they actually do. But I think the resources have been invaluable for COVID, what they provided. But go to long COVID news. So this was a paper that came out saying this is a Wire article. I was cut off there, but talking about immune cells. Pedro was, I don't think Pedro's here from our collaborator in Brazil, but he might be interested that since he's done a lot of work with monocytes. But there's this paper here. I put the link here, some couple of figures here. It's saying that monocytes could be one of the key suspects, and as they put in the title here in the article, that are key influencer of long COVID definition or long COVID behavior, or your outcome, I should say. And what they show, this is one of the figures they have here from figure 1 here. This is kind of showing it right at the acute phase, acute patients get to COVID. There's a distinct monocyte change or signature that happens and they look at immune-related cells, so this is probably related to the inflammatory.
Can someone explain what monocyte means? I've never heard the term.
Yeah, monocytes is one of the. It's basically one of the cells that are in your blood that floats around in your body. Related to a lot of immune function.
Okay. I
don't know if someone wants to give a better definition of it, but simply that's that. It's one of your circulating cells that's in your body, found in the whole blood. And it's involved. You want healthy monocytes in your body because that helps your immune system to be active and behave better. But yeah, go ahead.
I was just going to say one aspect of that, if you have low immune cells following COVID infection or long COVID, and then you have, say, problems with tumor development or cancer, you're not going to be able to clear them because your immune system is going to be dysregulated. So that's just one potential mechanism. Could be hypothesized as to how that could contribute to negative symptoms, but I'm sure there's others as well.
Yeah, that's very, exactly. I don't know if you others have comments on it. Feel free.
Yeah, no, this is Steve. Everything you've heard is right. Just in a bulleted way, these monocytes the cells they come from and then they themselves can develop many lineages depending on demand. In cancer, some can go in directions, and macrophages are another cell type that can give rise to cells that help tumors develop, cells that help quash tumors. So it's a very important system of cells that comes out of your bone marrow. Yeah.
They're also part of the innate immune system. So you have your adaptive immune system, which is where you produce antibodies and the B cell, the whole B cell T cell response. That's the adaptive immune system, and the monocytes are part of your innate immune system, which is the first reaction, the first part of the immune system to react to any kind of threat.
Oh, that's actually a really helpful explanation, because I learned all about the T cells because we actually helped bring the first T cell test to market. We helped do the trials and everything from beginning to end with adaptive biotechnologies.
So they cover in this. I didn't show the figures here, but there's some markers for example related. Yeah, there's some figures here. Markers they looked at in this paper related with immune innate activity. It's not here. You have to go look in the paper that Sigrid brought up. So you'll see some of the things that you talk about related to T cell activity and things like that, or indirectly related, I should say.
I found out yesterday. I found it fascinating. The company that developed the mRNA vaccine strategy for COVID in concert with Pfizer, small company, they had very little money. Now, obviously, they've got tons of money. So what have they done? They've gone into cancer. And for vaccine strategy and everything based on mRNA type deliveries. What can you do for vaccines? What can you do? It's just fascinating. And I probably recruited a lot of good people. They just have lots of money on hand. So that's really interesting. Yeah. Something else COVID spawned, I guess, research that could benefit a lot of areas.
I've read something about that, talking about maybe potentially applying the same sort of vaccine for cancer. It'd be interesting to see what happens.
Absolutely. Yes. They actually have a trial up right now for an antibody against a cell surface marker that the people in the breast field, that Dennis Slayman, also have a unique way, and all these trials are going on. Targets cell surface proteins. Yeah.
Oh, wow. Yeah, I didn't know about that. But yeah, this would be, I think, an interesting paper for folks to read, because it's basically talking about, compared to the healthy controls. It's kind of like what Pedro did. But in a sense, in Pedro's paper that we're all familiar with, he took healthy monocytes, infected them with SARS-CoV-2, and he started seeing from healthy individuals to people who are, insulin resistance, and people who are obese, people who are fit, so on. And then he started seeing some interesting changes happening in the monocyte population based on that. But this is actually taking monocytes from patients who have been infected with COVID, acute COVID, and then I think over time they took some other samples too. But yeah, basically they're pointing to that monocyte irregularity is going to be the key for long COVID. That's what they're claiming in this paper. It might be one of the things, might not be the key, but it might be one of the things. And there's some, they look at profiles, different inflammatory and immune profiles throughout the paper. So maybe for immune paper, you might want to cite this one, or inflammation paper, cite this paper in the discussion. Might be interesting to do.
As you often help us with, can you put it in chat?
Yeah, definitely. As we're going over, 20 seconds, I need to stop sharing for a second.
It's really interesting, yeah.
Stop sharing, throw this in the chat. I'll share the slides also, but yeah. Right. Here's in the chat. Let me go back to sharing. You guys could follow on the paper. Okay. This is another paper here. This article, this is just an article talking about long COVID rates are maybe higher in rheumatic diseases. So you can take a look at that. But this paper here I thought was interesting, looking at exercise tolerance is reduced. And we kind of knew this from anecdotal or epidemiological type evidence. They actually did some proteomics on patients, with long COVID. And one top thing that for a mitochondrial paper, you see OXPHOS is suppressed. So mitochondrial suppression, OXPHOS. They talk about, in the paper, they have 3 references to mitochondria. some of these markers, I'm sure Joe and Doug and other folks here recognize these as key mitochondrial markers. You can see here also geontology mitochondrial envelope pathway being regulated in these people who are they looked at for these long COVID patients. Their number for long COVID. Oh, sorry. Go ahead.
What tissue was this, or samples were they looking at?
They're looking at from long COVID patients that they just took blood plasma obtained.
Okay.
Venous blood plasma. What it is.
Huh.
So
there's some immune markers.
They didn't look at tissues. Yeah. Sorry. Go ahead.
I think there's some immune markers which came up in the RAS pathway as well, Steve.
Yeah.
I don't know if I think ANGP.
I tell you, I just came back from an intense research foundation meeting I participated in with great people. And Doug would be glad to know, Joe, mitochondria are starting to really get drilled down on in inflammation and immune. Not so much the details, but some of the RAS genes, they're even got inhibitors now. ZBP1, I'll talk to people about it. But just to say again, it's just spilling over, and it's great, I think, Doug and Joe would say, but it's not silent anymore.
Sorry, Steve, you said someone has an inhibitor for ZBP1?
They do indeed.
Was that Jerry Sladle?
It may have been. Yeah, we'll go into it. I didn't want to do
it now. Okay.
Just again, it's another example of how the COVID research is spilling out into these other fields. It's kind of
yeah, definitely. But yeah, this paper, they only have this one figure, so and they have tables for the patient population they looked at. But this one figure they did proteomics on, you can see they talk about metabolic reprogramming inflammation. Yeah, as Joe pointed out, there's some RAS, ANGPT2 and other ones. Serpin E2. I don't think we have that in ours, but yeah, we do have that, I think. Yeah.
Yeah.
But then
E1, E2. Yeah.
Yeah. And then they have some mitochondrial factors. I'm sure Joe recognizes.
Yeah, they got NOS, and some of the other immune factors can interact as well. So.
But it's a short little paper. They have a patient population. I forget what it is in here, but you can read the paper out, and you have to look at 21 patients. So it's a smaller population, but still, it's interesting to see all these factors. And then they related. They show that these patients actually did had a reduced tolerance to exercises for long COVID, and then trying to figure out what biologically they might impact them.
It's a very interesting question about the exercise with long COVID, because we looked in our lab at mitochondrial deficient mice actually improved from exercise, and some of them did and some of them didn't, which was unfortunate because sometimes the exercise with the mutation would actually have a negative effect. So this is good. This is good action. I'm glad we're on the right track for both the inflammation and the mitochondrial segments of covert. So
that's why I thought you guys would be happy to see this paper. I'm going to put the link in the chat for this one also, so you can.
The fact that they found it and they weren't even looking means that we're on the right track.
They weren't. They weren't. I don't think mitochondria. Maybe the mitochondria was on the radar. Who knows? You have to talk to them. But
okay.
but they sure showed either way. But that's interesting. Again, this is another paper for inflammation group. This is actually an interesting one because what they did was they did apheresis. The kind of the filtering of the blood. I'm not too familiar with it, but that's, I guess, what it does, right? If someone is more familiar with it, they could discuss it. But what they did, they looked at patients after they went through this therapeutic, after 2 cycles of therapeutic apheresis. And they saw that indeed, when they did this, I forget the patient population they had in this paper. So you have to go look at the methods to know what the N was for the patients. But these are the figures from the paper. What you see, they actually talk about oxidative factors. So they only looked at raw stuff. They didn't look at other mitochondrial things.
This is. I think this might match with the monocyte. I guess deficiencies.
Yeah, because apheresis. They could filter bad monocytes.
So apheresis is just a general term to filter the blood, basically. And so I donate double reds. Those are red blood cells, and they put back everything else. They just filter out the red blood cells. But they also do it with the platelets. So you can filter out the platelets and put everything else back in. You can filter out whatever, I guess they're filtering out autoantibodies, and I don't know what they're doing in this one.
Yeah, I didn't read the methods of it, so I'm not sure which one they're doing. Yeah, exactly. That's good, Steve.
what I've seen most is apheresis being used. They're trying to filter out the actual microclots, but I've been also told that that doesn't work.
Yeah. So again, the methods I didn't read. I didn't read this paper methods too detailed, so I'm not sure what they're doing the therapeutic part of it filtering, but they're filtering something out. Maybe it's what you're saying, Sigrid. They might just do a standard filter. I don't know. Platelets, maybe monocytes even. I don't know.
Well,
but look at the methods of the paper. I'm sorry I didn't lead in depth. But the main point of that is through the two therapeutic things is showing like reduction, as you see here, for inflammatory factors once they actually do that. They also see lipid levels are reduced. They see, things like the rheology reduced, which is that other paper, fibrogen or fiber fibrogen related activity. And then here was just neurotransmitters, not antibodies. You can see that. But this is, it's a short paper. They kind of looked at a few factors, as you see here. This is all the figures in the paper. But
it goes at Diana's question about whether it works or not. But there's the fibrinogen and all of this, and fibrin fibers, and you can't form clots without those.
Yeah.
So, it is part of the genesis of the clots. Whether the procedure works to take out the microclots, I have no idea.
Yeah, and you can see the pictures of the blood, measuring the blood, like the post and pre. You can see a nice healthy one before is that. So it's an interesting paper. Again, it's a short. I have to look at the N and what they filtered out, but they're relating. Yeah, so it's interesting here. Yeah, I don't know if people have any questions or comments on this one, but people are getting more and more clues with long COVID. A lot of it seems to keep confirming our hypothesis that Doug had initially. We all had about the mitochondria. Long COVID might be a mitochondrial disease, but more proof is maybe backing that up. This one I just didn't have time to put what's the article, but this is really just a summary of the article talking. I just put the link here that we've heard in the past months, papers coming out saying there's viral reservoirs left over, and people are potentially saying that could be the long COVID thing. These viral reservoirs, like Rob has shown, the spike, is in different organs after a long time after the acute phase, and other papers are showing that too in other organs. Whether it's replicating or not, that's I guess it's up in the air depending on who you look at. But this is a nice NPR article just talking about the different papers, summarizing with the papers that you've probably all seen already. So if people wanted to look at that. And then just going into the different variants, as I said, this XBB.1.15 is actually majorities, but you can see that 1.16, the one that started in India. This is that 1.9.1 is the one that started in Singapore. They're rising a little bit. But the good news is the wastewater is still showing decline. BioBot actually now updated that are actually showing you. Before they didn't have this distinction between 1.9 and 1.16 for the XBBs, but they're showing now. You can see a little more of that, which one's starting to show more prominent nationally in the U.S. for which new variants going over. But as you can see, for example, my county in Middlesex, like last week there was a little spike, but this spike is, you see these little blips coming up and down, and the wastewater overall trend keeps going down, as you see that nationally. So that's a good sign. Again, the variants, this is the CDC tracker in the US. You can see that again, so XBB.1.5 is pretty dominant, but you can see now, 1.16 here in the purple is starting to rise. 1.9.1 is also competing. But again, from what I understand, the mutations, the additional mutations that occur with these two are not too far apart from 1.9.1 and 1.16, if I'm not mistaken. And people have more knowledge about it, please jump in. But I don't think they're too far apart from what I understand for the mutations that occurred. But you can see the distribution you have in here. What variants, are 1.16. It seems to be taking over more than other ones. But as I said, the good news is doesn't seem to be increasing hospitalizations or causing any more symptoms other than the conjunctivitis that have been reporting as a new
It
symptom.
seems to be, maybe on your subsequent diagrams, it was lower last week when you talked about it 1.6. It's going up. But
Yep.
So has anybody picked it up yet in cases, actual cases? They must be if they're citing the.
Yeah, I mean, I think in the U.S. They have to be getting infected, so you see the cases,
Yeah,
and you can see that in the wastewater too. See,
yeah,
it's
to widen that pie chart thing. Yeah, they must be seeing it.
Yeah, I mean that's the thing. So there's been really no increase in hospital. So this is my last slide, so I don't have time this week to actually show an individual breakdown. But you can see still the hospitalizations are declining, even though, as you pointed out, Steve, the XBB.1.16 is going up. You know, as we see from the previous. Yeah.
So what's good for the country, or not good in the country, this meeting I just mentioned to you, I came back yesterday. Many immunologists and everything, I was talking to one, they're not even aware of 1.16 or Arcturus or whatever.
Oh, no? Yeah.
No. It's news to everybody, so I guess it's not everybody, but to people you would expect who used to follow it fairly closely.
Right.
It's the news is they don't follow it very much anymore.
Yeah, and that's since the WHO has now declared end of emergency. Maybe less and less people will follow, unfortunately. But I mean, the good thing is, I guess the positive news we could say, you could see. I mean, again, the new case report. This is pretty unreliable these days. Not just the U.S. but Europe, everywhere. Since there's a lack of the trackers are being shut down, so and there's a lack of good reporting happening. But if you look at the hospitalizations for the countries who do report that, that decline is happening. So that's what I guess that would be my indication.
What happened in India? Did you get a chance to look where it was? Has it already peaked and gone?
Supposedly it has, but there's no good reliable. So if I show the our world in data for India, it just shows a flat line since last June. They just stopped reporting, they stopped recording that. But when you look at what they discuss in the media, when they actually, the scientists there and the reporting that people do internally, I think it's gone up and it went down. Supposedly it didn't hit the first, the blip that you saw with, the big blip we saw, for example, here with the different Omicrons that popped up. That didn't happen for XBB.1.16. According to what the scientists have said there. So I mean, again, that's positive and I think they really say it's mainly because, the symptoms are not too far different. And I think one of the claims I've heard too is if you were infected with the XBB.1. The 1.5, you probably have still some immunity left over for these new variants, so, and since a high portion of people got infected with XBB.1.5 or a related one, they think, that could be it. I don't know. That's speculation, but that's it, I guess, for this week, unless people have any questions, any